Esteemed colleagues, today, we stand at the intersection of space exploration and cutting-edge biomedical science, poised to explore a critical area of astronaut health: spaceflight-associated neuro-ocular syndrome, or SANS. As we start on this journey, we are tasked with deciphering the complexities of how space travel impacts the human body, particularly the neurological and ocular systems. This challenge is not merely an academic one, it is vital for the future of human space exploration. In the pursuit of answers, it is essential to acknowledge the limitations we face. Initially, the prospect of contributing to a knowledge graph through integrated causality analysis was a tempting path. However, upon thorough evaluation of the available data, particularly in the context of astronaut health and SANS, I made a conscious decision to pivot toward an approach grounded in a deeper understanding of the data's limitations. In this context, we face a fundamental issue: causal inference. The studies currently available involve a limited sample size, with mice being the model organism. This small sample size, coupled with the high biological variability inherent to living organisms, undermines the statistical foundation required to make robust causal claims. To put it plainly, any model attempting to establish causality would lack scientific rigor, and thus, I chose to deliberately rule out this approach. In this instance, embracing causality would have been not just premature but scientifically inappropriate, risking misinterpretation. Instead, I chose to focus on a method that lays the groundwork for a more accurate understanding, one that can evolve as we gather more data and refine our approaches. The method I have opted for is complex network analysis, a powerful tool that can identify patterns and relationships that might otherwise remain obscured in raw data. While this approach cannot directly resolve causal questions, it provides an invaluable starting point. In my previous work applying complex network analysis to neuroscience, we achieved high accuracy in identifying clinical patterns. These results suggest that, with properly labeled data and refined methods, such approaches could in the future evolve into tools capable of modeling and predicting causal structures. Thus, my decision to focus on this alternative pathway was not a retreat, but a recognition of the need for a more robust, scientifically grounded framework that can eventually lead to clearer answers. The exploration of SANS is not just about understanding the symptoms but also about constructing the tools that will ultimately unlock the deeper mechanisms at play. An essential aspect of this approach is the role of gene expression data, particularly that which is precisely labeled and consistently identified. Without accurate SANS labeling, drawing causal connections between gene expression and SANS becomes speculative at best. This recognition reinforces my decision to move away from a causal-based methodology in favor of one that is based on identifying meaningful patterns within the data. While the data we have at this stage did not reach statistical significance, this approach represents a foundational step. We are not yet solving important questions, but we are building a framework that, with more data, could provide a more accurate integration of information in the future. Now, let's discuss the method itself, these complex networks. The initial graphs I have constructed are a modest beginning. Each original graph contains over 1 billion edges and more than 55,000 nodes. These actual graphs are merely a sample, constructed from clusters of genes that show the highest differential expression. While these graphs are computationally intensive and are only a preliminary step, they are crucial. They represent the first foray into a more comprehensive method that could, in time, evolve into a robust tool for understanding the network of gene interactions. The power of this method lies not in the immediate resolution of all questions but in its ability to highlight potential biological relationships, insights that may not be immediately apparent from the raw expression data alone. This approach can ultimately provide a blueprint for future work, one that may allow us to build more accurate models of spaceflight-induced health conditions, like SANS. However, due to the sheer computational complexity involved, the full potential of this method was not realized within the time constraints of this competition. These graphs represent only a fraction of what could be achieved with more time and more advanced computational resources. Nevertheless, these early results offer a glimpse of the power inherent in complex network analysis applied to biological data. As we look to the future, the path forward is clear. The next steps involve refining these methods, scaling up the graphs, and, critically, incorporating more data. The hope is that as we continue to collect more detailed, high-quality data, especially gene expression data with precise labeling, we will gain a deeper understanding of the molecular underpinnings of SANS and other spaceflight-associated health conditions. This could ultimately lead to more targeted interventions, allowing astronauts to return safely from their missions with their health intact. In conclusion, the journey to understand spaceflight-associated neuro-ocular syndrome is still in its early stages, and the complexity of the task is immense. But we are making progress. By focusing on complex network analysis, we are not just waiting for answers to fall into place, we are actively building the tools that will lead to those answers. Through patience, precision, and collaboration, we will unlock the mysteries of human health in space, one data point at a time. Thank you.